Biological classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdom was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organisms into plants and animals was early done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. Besides the gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, method of reproduction, evolutionary relationships, etc. Classification system for the living organism have hence undergone several changes over the time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups organisms be included under these kingdoms have been changing. The number and nature of other organic kingdom have also been understood differently by different scientists over the time. R.H. Whitaker 1969 proposed a five kingdom classification. The kingdoms defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 gives a comparative account of different characteristics of the five kingdoms. The three-domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdom in the third dormancy and thereby a sixth kingdom classification. You will learn about this system in detail at higher classes. Let us look at this five kingdom classification to understand the issues and consideration that influence the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperms, and the angiosperm under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae cyanobacteria with other groups which were eukaryotic. It also grouped together the unicellular organism and the multicellular ones. Say for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants. Through they also showed a characteristic difference in their wall composition. The fungi had chitin in their wall while the green plant had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom. Kingdom fungi. All prokaryotic organisms were grouped together under Kingdom Monera, and the unicellular eukaryotic organisms were placed in Kingdom Protista. Kingdom Protista has brought together Chlamydomonas chlorella earlier placed in algae within plants and both having cell walls with Paramecium and Amoeba, which were earlier placed in Kingdom which like Silval, it has put together organisms which in earlier classifications were placed in different kingdoms. This happened because the criteria for classification changed. This kind of changes will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities but is also phylogenetic, i.e., is based on evolutionary relationships. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whitaker system of classification. The kingdoms Plantae and Animalia, commonly referred to as plant and animal kingdoms, respectively, will be dealt separately in chapter 3 and 4. Kingdom Monera. Bacteria are the sole members of the kingdom Monera. They are most abundant microorganisms. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot springs, deserts, snow, and deep oceans where very few other life forms can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under four categories based on their shape: the spherical coccus plural cocci, the rod-shaped bacillus plural bacilli, the common-shaped vibrium plural vibrio, and the spiral spirillum plural spirillum. Though the bacteria structure is very simple, they are very complex in behavior compared to many other organisms. Bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic, i.e., they synthesize their own food from inorganic substrates. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, i.e., they depends on other organisms or on dead organic matter for food. Archaebacteria. These bacteria are special since they live in some of the most harsh habitats such as extreme salty areas halophiles, hot spring thermoacidophils, and marshy areas methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows and buffaloes, and they are responsible for the production of methane biogas from the dungs of these animals. Eubacteria. There are thousands of different eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as a blue-green algae, have chlorophyll A similar to green plant and are photosynthetic autotroph figure 2.2. The cyanobacteria are unicellular, colonial, or filamentous, freshwater marine or terrestrial algae. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in a specialized cell called heterocyst. Example: nostoc and anabaena. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia and use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers. Many of them have a significant impact on human affairs. They are helpful in making curd from milk, production of antibiotics, fixing nitrogen in legume, roots, etc. Some are pathogens causing damage to human being, crops, farms, animal and pets. Cholera, typhoid, tetanus, citrus canker are well-known diseases caused by different bacteria. Bacteria reproduce mainly by fission, figure 2.3. Sometimes under unfavorable conditions they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacteria to the other. The mycoplasma are organisms that completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. Kingdom Protista. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protistian to one biological biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, lemurs, and protozoans under Protista. Members of Protista are primarily aquatic. This kingdom form a link with the other dealing with plants, animals, and fungi. Being eukaryote, the protistian cell body contain a well-defined nucleus and other membrane-bound organelles. Some have a flagella or cilia. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. Chrysophytes. The groups include diatoms and golden algae dismids. They are found in fresh water as well as in marine environments. They are microscopic and float passively in water current plankton. Most of them are photosynthetic. In diatoms, the cell walls from two thin overlapping cells which fit together as in a soup box. The walls are embedded with silica and thus the walls are indestructible. Thus, diatoms have left behind large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oil and syrups. Diatoms are the chief producers in the ocean. Dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, or red depending on the main pigments present in their cells. The cell wall has stiff cellulose plates on the outer surface. Most of them have two flagella. One lies longitudinally and the other transversely in a furrow between the wall plates. Very often red dinoflagellates, example goniolex, undergo such a rapid multiplication that they make the sea appear red tides. Toxins released by such a large number may even kill other marine animals such as fishes. Euglenoid. Majority of them are freshwater organisms found in stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in the presence of sunlight, when deprived of sunlight, they behave like heterotrophs by predation on other smaller organisms. Interestingly, the pigments of euglenoid are identical to those present in higher plant example euglena figure 2.4b. Sleep mode. Sleep mode are saprophytic protist. The body moves along decaying twigs and leaves engulfing organic material. Under suitable conditions, they form an aggregation called plasmodium which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies bearing spores at their tips. The spores possess true wall. They are extremely resistant and survive for many years. Even under adverse conditions, the spores are dispersed by air current. Protozoans. All protozoans are heterotrophs and live as predator or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans, these organisms live in fresh water, seawater, or moist soil. They move and capture their prey by putting out pseudopodia false feet as in amoeba. Marine forms have silica cells on their surface, some of them such as enta-amoeba are parasites. Flagellated protozoans, the members of this group are either free-living or parasitic. They have flagella. The parasitic forms cause diseases such as sleeping sickness, example, trypanosoma. Ciliated protozoans. These are aquatic, actively moving organisms because of the presence of thousands of cilia. They have a cavity gullet that opens to the outside of the ciliary surface. The coordinated movement of rows of cilia causes the water laden with food to be stirred into the gullet. Example, Paramoecium, figure 2.4d. Sporozoans, this includes diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium malarial parasite which causes malaria, a disease which has a staggering effect on human population. Kingdom fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on a moist bread and rotten fruits. The common mushroom you eat and toadstools are also fungi. White spots seen on mustard leaves are due to parasitic fungus. Some unicellular fungi, example yeast, are used to make bread and beer. Other fungi cause diseases in plants and animals. Wheat rust causing Puccinia is an important example. Some are the sources of antibiotics, example penicillium fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in the refrigerator? Yes, it is prevent food from going bad due to bacterial or fungal infections. With the exceptions of yeast, which are unicellular, fungi are filamentous. Their body consists of long, slender, thread-like structures called hyphae. The network of hyphae is known as mycelium. Some hyphae are continuous tubes filled with multicellulated cytoplasm. These are called conocytic hyphae. Others have septae or cross wall in their hyphae. The cell wall of fungi are composed of chitin or polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from dead substrates and hence are called saprophytes. Those that depend on living plant and animals are called parasites. They can also live as a symbiont in associations with algae as lichens, with root of higher plant as mycorrhiza. Reproduction in fungi can take place by vegetative means, fragmentation, fission, budding. Asexual reproduction is by spores called conidia or sporangiosperms or zoospores, and sexual reproduction is by oospores, ascospores, and basidiospores. The various spores are produced in distinct structure called fruiting bodies. The sexual cycle involves the following three steps. First, fusion of protoplasm between two motile or non-motile gametes called plasmogamy. Second, fusion of two nuclei called karyogamy. Meiosis in zygote resulting in haploid spores. When a fungus reproduces sexually, two haploid hyphae of a compatible mating type come together and fuse. In some fungi, the fusion of two haploid cells immediately result in diploid cell 2n. However, in other fungi, Ascomycetes and Basidiomycetes, an intervening dikaryon stage n plus n, i.e. two nuclei per cell, occurs. Such a condition is called a dikaryon and the phase is called dikaryophase of fungus. Later, the parental nuclei fuse and the cells become diploid. The fungi form fruiting bodies in which reduction division occurs, leading to formation of haploid spores. The morphology of mycelium, mode of spore formation, and fruiting bodies form the basis for the division of the kingdom into various classes. Phycomycetes. Members of Phycomycetes are found in aquatic habitats and on decaying wood in moist and damp places or as obligate parasites on plants. The mycelium is aseptate and quinocytic. Asexual reproduction takes place by zoospores motile or by aplanospores non-motile. These spores are endogenously produced in sporangium. A zygospore is formed by fusion of two gametes. These gametes are similar in morphology, isogamous or dissimilar, anisogamous or isogamous. Some common examples are Mucor figure 2.5a, Rhizopus, the braid mold mentioned earlier, and Albuga, the parasitic fungi are mustard. Ascomycetes, commonly known as sac fungi, the ascomycetes are mostly multicellular, example penicillium, or rarely unicellular, example yeast. Saccharomyces, they are saprophytic decomposers, parasitic are coprophilous, growing on dung. Mycelium is branched and septate. The asexual spores are conidia produced exogenously on the spatial mycelium called conidiospores. Conidia and germination produce mycelium. Sexual spores are called ascospores, which are produced endogenously in sac-like asci similar ascus. These asci are arranged in different type of fruiting bodies called ascocarps. Some examples are aspergillus, figure 2.5b, claviceps, and neurospora. Neurospora is used extensively in biochemical and genetic work. Many members like morel and truffles are edible and are considered delicious. Basidiomycetes. Commonly known forms of Basidiomycetes are mushroom, bracket fungi, or puffball. They grow in soil, on logs and trees stumps, and in living plant bodies are as parasitic. Example rust and smut. The mycelium is branched and septate. The asexual spores are generally not found, but vegetative reproduction by fragmentation is common. The sex organs are absent, but plasmogamy is brought about by fusion of two vegetative or somatic cells of different strain on genotypes. The resultant structure is dikaryotic, which ultimately give rise to basidium. Karyogamy and meiosis take place in the basidium, producing four basidiospores. The basidiospores are exogenously produced on the plasmodium. Plural basidia. The basidia are arranged in fruiting bodies called basidiocarp. Some common members are agaricus mushroom, figure 2.5c, Ustilago smut, and Puccinia rust fungus. Neutromycetes, commonly known as imperfect fungi because only the asexual or vegetative phases of these fungi are known. When the sexual forms of these fungi were discovered they were moved into classes they rightly belong to. It is also possible that the asexual and vegetative stage have been given one name and placed under Deuteromycetes, and the sexual stage another and placed under another class. Later, when the linkages were established, the fungi were correctly identified and moved out of the Deuteromycetes. Once perfect sexual stages of membranes of the Deuteromycetes were discovered, they were often moved to a Ascomycetes and Basidiomycetes. The Deuteromycetes reproduce only by sexual spores known as conidia. The mycelium is septate and branched. Some members are saprophytes or parasites, while a large number of them are decomposers of litter and help in mineral cycling. Some examples are alternation, coletrichum, and trichoderma. Kingdom Plantae. Kingdom plant included all eukaryotic chlorophyll containing organisms commonly called plant. A few members are partially heterotrophic such as the insectivorous plant or parasites. Bladderwort and Venus flytrap are examples of insectivorous plant, and Cascata is a parasite. The plant cell have an eukaryotic structure with a prominent chloroplast and cell wall mainly made of cellulose. You will study the eukaryotic cell structure in detail in chapter 8. Plant T include algae, bryophyte, and pteridophyte, gymnosperms, and angiosperms. Life cycle of plant has two distinct phases: the diploid, so sporophytic, and haploid gametophytic that alternate with each other. The length of haploid and diploid phases and whether these phases are free living or dependent on other vary among different groups in plants. This phenomenon is called alternation of generation. You will study further detail of the kingdom in chapter 3. Kingdom Animalia. This kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cell lack a cell wall. They directly or indirectly depend on plant for food. They digest their food in an internal cavity and store food reserves as glycogen or fat. Their mode of nutrition is holozoic. By ingestion of food, they follow a definite growth pattern and grow into adult have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanisms. Most of them are capable of locomotion. The sexual reproduction is by copulation of male and female followed by embryological development. Silent phyla of various phyla are described in chapter 4. Viruses, viroids, prions, and lichens. In the five kingdom classification of Whitaker, there is no mention of a lichen, and some acellular organisms like viruses, viroids, and prions. These are briefly introduced here. All of us who have suffered the ill effect of common cold or flu know what effects virus can have on us. Even if we do not associate it with our condition, viruses did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the body living cell. Once they infect a cell, they take over the machinery of the host cell to replicate themselves. Killing the host would you call viruses living or non-living? Virus means venom or poisonous fluid. Dmitry Ivansky, 1892, recognized certain microbes as casual organisms of the mosaic disease of tobacco, figure 2.6a. These were found to be smaller than bacteria. Because they passes through bacteria-proof filters, M.W. Banerjee 1898 demonstrated that the extract of the infected plant of tobacco could cause infection in healthy plant and named the new pathogen virus and called the fluid as Contagium vivum fluidum, infectious living fluid. W. M. Stanley 1935 showed that viruses could be crystallized and crystals consist largely of protein. They are inert outside their specific host cell. Viruses are obligate parasite. In addition to protein, viruses also contain genetic material that could be either RNA or DNA. No virus containing both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plant have a single standard RNA and viruses that animal infect have either single or double standard RNA or double standard DNA. Bacterial viruses are bacteriophages. Viruses that infect the bacteria are usually double stranded DNA viruses, figure 2.6b. The protein code called capsid made of small subunits called capsomers protect the nucleic acid. These capsomers are arranged in helical or polyhedral geometrical forms. Viruses causes disease like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by a virus. In plants, the symptoms can be mosaic formation, leaf rolling and curling, yellowing and vein clearing, dwarfing, and stunted growth. Viroids. In 1971, Teodiner discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein code that is found in viruses, hence the name viroid. The RNA of the virus was of a low molecular weight, prions. In modern medicine, certain infectious Neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. The agent was similar in size to viruses. These agents were called prions. The most notable diseases caused by prions are bovine spongiform encephalopathy, BSE, commonly called mad cow disease in cattle, and its analogous variant, Jacob disease (CGD) in humans. Lichens. Lichens are symbiotic associations, i.e., mutually useful associations between algae and fungi. The algal component is known as phycobiont and fungal component as mycobiont, which are autotrophic and heterotrophic respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their associations that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. Lichens are very good pollution indicator. They do not grow in polluted areas. Summary. Biological classification of plants and animals was first proposed by Aristotle on the basis of simple morphological characteristics. Linnaeus later classified all living organisms into two kingdoms, Plantae and Animalia. Whitaker proposed an elaborate five kingdom classification, Monera, Protista, Fungi, Plantae and Animalia. The main criteria of the five kingdom classification were cell structure, body organization, mode of nutrition and reproduction, and phylogenetic relationships. In the five kingdom classification, bacteria are included in kingdom Monera. Bacteria are compolitian in distribution. These organisms show the most extensive metabolic diversity. Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista include all single-celled eukaryotes such as chrysophytes, xenoflagellates, euglenoids, lymph nodes, and protozoans. Protistians have defined nucleus and other membrane-bound organelle. They reproduce both asexually and sexually. Members of kingdom fungi show a great diversity in structures and habitat. Most fungi are saprophytic in the mode of nutrition. They show asexual and sexual reproduction. Phycomycetes, Jascomycetes, Basidiomycetes, and Deuteromycetes are the four classes under the kingdom. The plant include all eukaryotic chlorophyll-containing organisms. Algae, bryophyte, radiophyte, gymnosperm, and angiosperms are included in the group. The life cycle of plant exhibit alternation of generations, gametophytic and sporophytic generations. The heterotrophic eukaryotic multicellular organisms lacking a cell wall are included in the kingdom Animalia. The mode of nutrition of these organisms in holozoic, they reproduce mostly by the sexual mode. Some acellular organisms like viruses and virids as well as lichens are not included in the five kingdom system of classification. Thank you.